Interaction Root Benefit series for Working Group 2. And also a big thanks, of course, to all of the speakers who were also very enthusiastic to fill in the program so nicely. So in this webinar series, of course, our primary goal is to get to know each other, so all of the different people involved in the action, and also, of course, to establish some future collaborations. So the format is going to be bi-monthly, so at the end of each month, mostly it's going to be the last Friday, we will join together for this meeting. And then, of course, what we want to do is we want to have our speakers give a very global overview of their lab's unique research expertise and our facilities. Every session is expected to take approximately 1 hour, so I'm going to keep it very brief in order to give the floor to the speakers as soon as possible. We have 2 speakers each session with a presentation of 20 minutes and 10 minutes provided for questions. And then, of course, also the talks will be recorded, with permission of the speakers, of course, and they will be posted on YouTube as well, so they can be watched afterwards as well. And then, of course, as you know, how you can join the Working Group 2 room because you all did it so successfully. Now, if you all have any questions, you can always contact Rafa or myself. Also, just a gentle reminder that this is what the program is currently looking like. So we have a lot of nice speakers filling up the program, but today we're getting started with Professor Sofie Goormachtig as well as Dr. Hannah Koch. So without further ado, let's get started and let's introduce our first speaker of today, Sofie Goormachtig. So Sofie Goormachtig is a UGent Professor and the PI at the VIB-UGENT Centre for Plant Systems Biology. Sofie focuses on how interactions between plant roots and neighboring organisms positively influence plant growth. And initially, the emphasis was on the symbiosis between legumes and rhizobia. She studied the interaction between the tropical legume Sesbania rostrata and the bacterium Azorhizobium caulinodans, and unravelled the early signaling events and specific adaptations that have evolved to enable nodulation upon water submergence. Next, our group studied long-distance control of nodule organogenesis in the model legume Medicago truncatula. Her group currently takes great initiatives in order to introduce soybean as a new crop in Flanders. Research extends beyond leguminous plants as well. So the team combines root microbiome analysis with proteomics, transcriptomics, genetic approaches, and high throughput phenotyping to elucidate the mechanisms by which microbes communicate with plant roots, resulting in beneficial effects for the plants. She has a strong interest in the validation of basic science and has contributed to the startup companies Afea Bio as well as Protealis. So, Sofie, the floor is yours. If I think Rafa is putting the presentation now.
Yeah, Rafa has to get something in and out. I can already change slides.
That's already good.
No, not anymore. There we go. I cannot change slides yet. Yeah, now I can. Okay. So hello, everybody. As Naomi said, I'm Sofie Goormachtig from the Center of Plant Systems Biology. Now, before we introduce what we do in the lab, I just want to say that, well, we are coming from Ghent, and Ghent is here somewhere in Belgium. So we are coming from this little country, a country which is very often known by the fact that there is something like, although it's small, there's even 2 smaller regions, and there is a third, even more smaller region. And we are very famous in the previous times that there were actually these fights between these 2 language regions in Belgium. But this is actually all politics, so I would not say too much about it, because what we're really famous for is, you should actually read in the Asterix book about Asterix in Belgium. And then you're going to get to know that we are a group, that we are people, the Belgians, who actually like foods. And we are also famous for the beer, the pralines, and, for instance, the French fries. Okay, now if you look to our institute, and we usually say we are the VIB-UGENT Centre of Plant System Biology. That means that we are apart from the university, Ghent University, and the VIB. Now, for many people, this is a kind of strange, because VIB is an institute of biotechnology, but we are actually also connected to the university. Now, the VIB originally has been established from labs of the university and was called an inter-university institute. So that directly tells you why we have always the both connections together. So we are living in Ghent. Ghent is a very nice medieval city, and the institute is a little bit outside of Ghent, but yeah, 5 kilometers biking is still very much okay. Good. So the VIB is then, as I said, an inter-university. So there are also the university, for instance, Leuven, there are other Antwerp, so there are other universities connected to the VIB in Flanders, and they cover different aspects of biotechnology. Now, most of those are actually has to do more with medicine, as you can see here, inflammation, cancer, and so on. But there's only this one institute which covers plants, and this is the institute we belong to, the plant systems biology. There's also a microbiology institute, and there's recently also one on AI. So altogether, we do the science in our lab. Then in the plant systems biology department, we are 19 research groups, and here you see the different of my PI colleagues running their own research groups covering different aspects of plant science, and in total we are about 330 co-workers. So if we work at VIB, I mean, they basically have this deals with the Flemish government, and what is very important in our science is that we should do curiosity-driven research, but link it to application. That also means that we do fundamental science, as many of us do, but that we do have to connect it somehow with excellence in entrepreneurial biotech. This means, of course, they don't expect from us to also be businessmen or women. That's not the idea, but we do have a whole group of people in the headquarters that support us in identification of applications from our basic research. This can be in terms of patents that you make, but also spin-offs. So this is actually, we have like definitely 30 people helping us for that. Now, today I'm going to present the rhizosphere, so the group I'm leading. And you see already Naomi here. Naomi is part of the group. This is a picture of us a couple of weeks ago on the New Year's party. And so you can also go to our quite new website to know who is doing what. So I will not really say who is doing what during my presentation, because I'm always afraid I will miss one, and then an opposite it might take a bit too long. But go to the website and then you're going to see who is here and who is doing what. Now, what do we do in the lab? Well, our lab covers PGPR biology, and I don't think I have to explain to you audience here what PGPRs are. I think you also know that PGPRs, they are involved in biocontrol, biofertilization, and biostimulation. And it's not all, we only do a little bit in biocontrol, but the most that we do in terms of PGPRs, and I also consider now the rhizobia with it, is about biofertilization and biostimulation. The research questions we attack in the rhizosphere group, generally very overarching, is how do these PGPFs impinge on plant growth and stress regulatory mechanisms? And by this knowledge, how can we identify mostly smart selection of strains for application? So we work on nodulation and mycorrhization. I will come to that later, but we also do biostimulation. Now, in consent from the PGPRs, we mostly work on, or we try to work on endophytes, so microbes that can colonize the insides of the plant roots. Now, I'm going to give you a couple of examples what we do, because that's the idea of the presentation here. So one of the aspects I would like to focus on is, for instance, the one about microbiome-based pipeline we have for the identification of PGPR. This is a project that has been started already more than 10 years ago, and a project that links our research to application. It's basically so it's by comparison of microbiomes that we look to identify, that we try to identify microbes, which come together with the isolation of the microbes, and then we try to get them into application. So this led already to 2 spin-off companies. We have Afea Bio, which is from 2017, so they almost reached their 10 years, and more recently then also Protealis, which is more about legume nodulation and legume breeding. Now, this microbiome-based pipeline, which is based on microbiome analysis, isolation, and then also phenotyping, we also use in the lab to address certain questions, to get certain patterns. And we have been working on cold stress in maize and lettuce, drought stress in maize, salt stress in tomato, and then also recently nodulation in soybean. Okay, here you see an example of a project that was a very applied project that Antoine has been doing. Basically, we went to the farmers, we talked about the local problems we have. We came to, for instance, cold as a main problem for lettuce growth in wintertime, and then we basically were looking for microbes that would help the growth of the plants during cold seasons. And then we identified a couple of microbes here who could really do the job, and this then turned into also application. Now, so from all this microbiome-based analysis that we have been doing and isolations that we have doing, during the time, then the basic science comes back in, and then we, at a certain point, we thought, these streptomyces are really interesting. They're interesting in terms of application because you can make spores, so they're actually nice for application, but we do not know much about how they act. We know a lot about them. We know that they are in many microbiomes. You can find them everywhere in soils. They're gram-positive. They're known to make a lot of metabolites. They're often growth promoting, and they help with abiotic stress responses, biocontrol, sometimes even biopesticides. They can isolate from them. And grains have this advantage of making spores. So we also started then a fundamental research project on a couple of streptomyces we have been isolating. And on the Munchgeberg, in a couple of months, I will explain you more in detail, but we have a couple of streptomyces that really help plants to grow when it's in dry conditions. So plants look much more healthy. Here you see an example of maize, and here you see how the weeds get straight, stay straight when you give them no water anymore. Now we then move to Arabidopsis. So in our department, not in the lab, but in our neighbors, as I just told you, we are with 19 PIs. There is a lot of knowledge about growth of Arabidopsis and growth processes in general. So what we then do is that we use their tools, what they have been, which kind of expertise they have, in order to understand how this growth promotion that we know from streptomyces, how does that really happen? So for this, we also move to Arabidopsis, and these streptomyces also induce growth on Arabidopsis. And by doing detailed phenotyping, it's not only measuring fresh weight, but number of leaves, looking into the leaves where they have more cells, bigger cells, doing really phenotyping in depth. We do our research. And then, for instance, for the case of streptomyces, this is something we are currently trying to publish, and so it's almost ready for publication. We see that at young stages, these Arabidopsis have more leaves, bigger leaves, more cells in the leaves, but it's actually what we see is a faster development. And at the time the plants start flowering, because we do see early flowering, we see that the number of leaves and cells per leaves actually start to equal out. So really showing that this streptomyces causes an acceleration in development rather than a true growth promotion, in that sense that the plants stay bigger compared to the control. Now, based on this, then we dig into literature, and in this case here, we are now trying to understand how this growth promotion is related, for instance, to SPL genes, which are known to be involved in the different aspects we have been studying. Now, so this is for the streptomyces. So it's really linked to phenotyping, and we have a lot of expertise here in the house for phenotyping. Then, of course, we also work to the endosymbiosis. That's our history. Maybe it has been less during the time, and now it gets really back into focus. And we work on mostly nodulation, but also have an arbuscular mycorrhization project. And so the target plants we have is tomato and currently mostly soybean. So for the tomato science, we, for instance, Naomi, and I guess she might also give you more insights later on in meetings, for instance, in the Munchgeberg. But there have been a lot of effort done in the lab to make a single nucleotide transcriptome map of tomato roots enriched in arbuscle containing cells. So we basically, via single nuclei, get the arbuscle containing cells. We could confirm this by known markers, but we also found new genes there in this cluster here. And as we knock them out, it seems that we really we miss the arbuscle. That's what you see here. So we really find new genes involved in the process. So we do have this big data set, and for everybody who is like with Juan Antonio, we already collaborate. So, but if you have interesting genes, we are definitely open to look how your genes perform in our data sets. Now, the soybean project is actually a project that also comes more from a societal and an environmental challenge that Europe, but also Flanders is taking. And this is that our agriculture is going hand by hand. So you see again here, the problem really comes from the application, and this is typically VIB, or no, more and more VIB, that we basically have to work on problems that are relevant. And this is all by the fact that in our current agriculture, so we all know we have to use less chemical and organic fertilizers because of the nitrogen problem. We, in our diets, we should shift more to plant protein and eat a bit less of animal protein. And within this whole challenge that I think Europe has to take is legumes come, of course, back into the picture. This is old knowledge, everybody knows this. And then soybean might come in here as well in Northwestern Europe. Why? It's an established crop, and getting it produced to northern latitudes would, for instance, allow Europe to make or grow more soybean themselves and be independent of other regions. Okay, now if you want to get a soybean, a new crop in a region, in a new region, like in Flanders or in Northwestern Europe, you basically first of all have to breed new varieties. And this is done by our partners, or our partner institute ILVO, and I've seen that there are some representatives of ILVO here now in the talk. And so they have been breeding for new varieties for which are adapted to our local conditions here. But it's as important to also have brady strains that are adapted to our regions. And also our ILVO colleagues, they knew that commercial inoculants were actually not doing very well here in May when it's still very humid and cold. So basically we started a big project here so first of all, well, if you want to do basic science that contributes to actually this challenge, you first of all need a good plant. And from all the breeding efforts, we have selected one variety which is adapted to here. We actually started to make the genome sequenced, and we also have a transformation platform in the lab that allows for genetic modification of soybean. So also gene editing is established. We also have hairy roots established. We can also do fix, so we can actually do many things with many molecular tools we have already for this variety, which can also grow here on our fields basically, and is adapted to our conditions. Now, we also need to have a good inoculum. And as I told you already, we knew from the previous works of our ILVO colleagues that this was a problem with the commercial inocula. And that's why I think it's already 3 or 4 years ago, we started a huge citizen science project in which we basically tried to look for a rhizobium that can nodulate soybean in Flanders here, and we did that by asking the citizens to grow a soybean in the hope we get nodules. We isolate the strains from the nodules, and like this actually get an inoculum which would work good in our regions. So this was a very nice project. We reached out to 1,200 people in Flanders. They grew soybean. I'm not going to go into detail here, but we could isolate some strains, and 1.5 years later, without really big rocket science such as microbiomes, we actually went already to the field and could isolate some strains that worked pretty well. Now, besides getting this application, we also get a lot of insights into the biology which we currently are digging in. Now, you see already this is a project where we involve stakeholders, and this is also something I think very important for our VIB. We do try to reach out not only to citizens, but also to farmers. So we now have also a big project where we grow soy in 50 fields in Flanders. But we also push to do something with this and to also reach out. And just to give you an example, the students from the group, they won this Nature Award this summertime with a movie they made about the use of PGPR. You should all have a look at it. And they also won money, but actually they really put the whole PGPR team on the map, also for people working in medical fields. And they also have a podcast. You can find that on not YouTube, the other one, which I now forget. Sorry. But anyway, it's a podcast the students are doing, and you can learn a lot about microbes and their science by listening to this podcast. Okay, so then of course I did not talk yet about the basic research we do on soybeans. So next to the fact that from the citizen science project we can ask many ecological questions there, we also dig more into the fundamentals of the nitrogen fixation process itself, and we now try to really dig more. We used to do more compatibility and knot factors and so on in Sesbania a long time ago. We also did autoregulation, which we still do a bit, but we also try to dig more into these carbon-nitrogen exchanges and how this relates to the yield we get into the plants. Now, the expertise we have at the molecular level, we do have a lot of proteomic approaches that we use. We have knowledge on shotgun proteomics, phosphoproteomics, ubiquitinomics, and also interactomics. This is not only in our lab, but also by actually collaborating with our PI colleagues here in the Institute. So we really can help people if you have questions related to these topics. And what we also do at the moment is making a map from soybean nodulation, where we superimpose the single nuclei transcriptome on spatial metabolomics, and we also try to put spatial transcriptomics on the map. So we are currently making our own maps. The single cell is on analysis, so we actually have all the data in. The metabolomics is also okay, but we still somehow have to work a bit more on the spatial transcriptomics. And in the future, we hope also that we can go more to the single cell proteomics. This is in collaboration with our colleagues from the lab. And of course, once we have this map, we can add perturbations and maybe then try to understand how this all works. Okay, so this is the end of my presentation. This is really in a nutshell what we are all doing. Of course, we don't do alone. We have a couple of main collaborations from VIB, but also outside VIB. And we are always open for helping people out if we can help them out. So feel free to contact us. So thank you. So then I don't know what I have to do now. Listening whether I have a question. Yeah, Toon, it's Spotify. The podcast is on Spotify.
Okay, thank you, Sophie, so much for your presentation. So if any of you have a question, or you can let me know. I switch on the camera of you, whatever you want. So just try to don't do at the same time. So if you have any question, you can switch on your camera and do it. I hope there is some questions.
Yeah, it's more an overview.
Yeah, of course. Now me. Okay. Well, I think everything is clear. Yeah, as you said, it's okay. We have one person with. So, yeah, as you said, it's an overview of the Benoit to give you a question, I think.
Benoit.
Okay. Hi, Benoit. No, we cannot hear you. So, Benoit. Let me check. Okay, now. Oh, I think you cannot. So maybe you.
No.
I think he can make the question through the chat.
Yeah, I think he joined the chat just as
a listener.
You joined just as a listener, and you cannot know. we okay. So yeah, Benoit, you need to write down the question. Sorry. No. Okay. Yeah, Benoit is writing.
Ah. Thank you.
People is writing, so maybe for the next time we can just join all of us as speakers too, and switch off the microphone. Okay. You can see the question, right, Sophie?
Yeah, yeah. But Judith is there for the spatial metabolomics.
Yeah.
Can Judith talk? I don't know. Judith is the person doing.
Yeah, maybe.
Ah, yeah, she cannot talk. Yeah.
Yeah. So this is something we need to.
Judith is coming to me now, so this is actually a lucky thing that we have with with a lot of people from our lab being involved now. Yeah, Judith.
Yeah, the point is you cannot now, if you join the platform as a listener, you cannot now speak. So sorry for that. Something that we are using a new platform, so we didn't know that before. So for next time, we know already. Sorry.
For the spatial metabolomics,
Yeah.
we're working with the VIB. You hear me, yeah? With the VIB spatial metabolomics score, and we're doing for the moment MALDI, so MSI, and we can practically detect every metabolite that you want. So for the moment, we're focusing on the primary carbohydrates, the TCA cycle, but also, we can do a lot of them.
There's a limit, Benoit, because sometimes you're not sure which peaks belong to which molecule. So there is definitely a limit, but the expertise is in the VIB headquarters, in one of the VIB groups, and they tell us then if we ask metabolites what they can and what they can't.
Yeah. Also, lipidomics is also possible with the spatial.
Yeah, also the lipidomics.
Was that the question?
Yeah, so more people is definitely. Okay.
Yeah, but they can also do amino acids, for example, or nucleotides. So they can do different. At which resolution? It's definitely better than the previous ones, but I have no clue. I think maybe Toon knows this. The resolution. Yeah. Like what we can definitely see in a nodule, you can definitely, if you look for a very small nodule, like an immature nodule, I'm just thinking that everybody knows how big a nodule could be. Then you really see like the difference in the vasculature and also inside the nodule.
Infected, uninfected cells, we also see. Have we tried arbuscle-containing cells? That's the headache for spatial metabolomics.
Yeah, you need to do cryo sections. So for the moment, this is we tried a lot. Naomi and Toon tried a lot, and it was not working.
Because of the sections that we cannot get clear.
10 micrometer. See, it's really a teamwork here. Eh? We all work together.
Okay.
Yeah, that's really. I'm still talking. Yeah, Naomi.
But we have still through Twinkelman, we have a special pipeline you follow for isolation. With streptomyces, we use special media, which is adapted to streptomyces.
Okay, I think now we can move on, right? Thank you all for the question. I'm sorry, we didn't know about you cannot speak if you join the, the platform as listener only. So we know for the next time. Sorry for that. So yes, you need to go in and switch off your microphone for next time, okay? Sorry about that. And then, so now it's turn of Hannah, but now me will introduce Hannah. So
I get that. I do now this. I stop mute and yeah.
me too.
So yes, indeed. Thank you, Sofie, for the very nice talk. And we move on now to the second speaker, which is Hannah Koch. So Hannah studied microbiology.